Good afternoon, everyone. Welcome. Thank you once again for joining us. This is it. This is our last presentation of Space Fest 2021. Thank you once again for joining us for all of these fascinating talks with scientists and researchers. And I'm happy to say that our last talk of the day is with some of our very own graduate students at the University of Minnesota. I'm Amber, the Public Science Events Manager here at the Bell Museum, and I want to first and foremost thank Ruth and John Huss, who help support Minnesota's astronomy classroom, and that's what helps us bring you all of these fascinating astronomy programs. So let's get started with our last and final program. Today, we're actually joined by two graduate students. They are Taylor Price and Teresa Mccarrell, and they are graduate students in geomicrobiology, and they have a special interest in astrobiology. Along with other researchers at the University of Minnesota, they study subglacial microbes, and I know they're going to tell you all about that. We can learn a lot about astrobiology by understanding these subglacial microbes, and Taylor and Teresa are here today to tell us more about their research. Ladies, take it away.
All right, thank you, Amber, for that kind introduction. I'm going to go ahead and share my screen now. Can you let me know if you see my screen?
We can indeed.
Awesome, great. So as Amber said, we are going to talk to you a little bit about astrobiology and our interest of that subject on icy moons. So with that, we'll go ahead and introduce ourselves. So as Amber said, we are graduate students at the University of Minnesota, and we study geomicrobiology. And today we'll call ourselves TNT. We're going to blow you away with astrobiology and geomicrobiology. So I'll go ahead and let Teresa introduce herself.
So I'm Teresa Mccarrell. I'm a first-year graduate student in the Hamilton Lab. And in the summer of 2019, I was able to work with samples of microbes that were taken from beneath the ice sheets of Antarctica. And my picture is the one on the left.
Yep, and then I'm Taylor. I work on microbes and glacier streams, as you can see on the right. And this is a picture of me working on a glacier in Grand Teton National Park. And so we just wanted to start off. I bet you guys are wondering what astrobiologists look like. Well, we're aspiring to one day be astrobiologists. So if you're wondering what that looks like, we're trying to do that, and you can also look like that too one day. And we're going to take you to the field and take you on a sampling trip, but first introduce to you what we do. So with that, we wanted to start off with asking you this simple question. Can you identify these planets? I'll just give you a couple seconds to think about this. Well, if you're feeling a bit confused or a little wondering if there were new planets discovered in our solar system, don't be alarmed. These are actually microbes, and I'll let Teresa tell you about them.
Yeah, these are actually pictures that I took myself through a microscope. So each dot is a colony of microbes, which means there are tons and tons of them on each single dot. And they come in this wide variety of colors, and these are all from beneath the ice in Antarctica.
Yep, and so we wanted to start off with this picture of microbes from Antarctica because it's a great segue into what astrobiology is. And so while these microbes may look like planets on these plates under the microscope, they're actually, as Teresa said, they're microbes from Antarctica. And we can study these microbes in extremely cold environments as a way to figure out what life, if we find life, may look like on icy moons and icy bodies in our solar system. And so what sort of icy bodies are candidates for life in our solar system? So these are moons such as Enceladus and Europa, and our neighbor Mars. And so these icy moons are targets for life because they're covered in a thick outer shell, as you can see in this picture, of ice that may harbor liquid underneath. And we need liquid water to support life, such as what we have here on Earth. And so on the right here, this is Mars, and Mars has these ice caps that form. And there's a lot of research right now trying to figure out if there is subglacial liquid on Mars to support life. But right now, there's a lot of research focusing on these icy moons of Jupiter and Saturn to understand if there may be water in these subglacial habitats. So how do we start to study life that may have evolved there or exist there? And what do these habitats look like out in our solar system? So these are pictures of the two moons that I just introduced on the previous slide, and this is Mars. And so if we were to try and understand these habitats, we'd have to start to think about what life looks like here on Earth. And so that's our main focus here, is astrobiology. And so astrobiology is the study of life that may exist on our other planets by studying life here on Earth. So in order to understand what icy life may look like on these moons, we have to go to the extreme environments here on Earth. So that means we're going to go to a glacier, and we're going to go study how these microbes exist and live in the glaciers here. And so these extreme habitats are analogues. This is the word that we use when we're talking about habitats that may support life or may look like habitats in our solar system that support life. And so if we were to go to one of these icy moons, this is Enceladus, this is Saturn's moon, we would see that there is an icy exterior. And if we were to crack open Enceladus, we'd see that there's a thick layer of ice, but underneath of that, there's liquid, and that's salty ocean. Underneath of that, we would see a sediment layer. And so by studying what similar habitats here on Earth, we can start to understand what sort of life lives in these salty oceans on Enceladus. In this picture, there are these volcano-looking things. And we're not going to talk about those today. They're really interesting. If you would like to learn more, you can go to NASA's website and learn about the current research that's going on with them. But I'm going to turn it over to Teresa now so she can tell you what those analog habitats look like here on Earth.
Well, before I start talking about the analog habitats, I'd like to call back right quick to that image previously shown here in the background. This is a Martian ice cap. So just like Earth, Mars has these ice caps. And just like how there are lakes beneath some of the ice in Antarctica, we think there could be lakes beneath some of the ice on Mars too. So now I'm going to transition to telling you a little bit about what's beneath the ice. So the image on the top is the outside of a glacier, and the image on the bottom is a cross-section of a glacier. So the very top layer is snow, and beneath the snow, we get to see some of these ice. And there's tons of different ice layers that are deposited over time. So older ice will be found deeper. And what can we find beneath the ice? Well, sometimes we find liquid. And sometimes this liquid is even a brine, which means it's salty, just like the waters beneath the icy moons. So that liquid and the sediment below the liquid is where we're trying to sample microbes from. And this is the tool that has been used to sample it. It's called the ice mole, and it uses its superheated tip to burrow through the ice, keeping the tip sterile with that heat, as well as continuing to melt the ice as it plows through until it gets to the bottom. And it collects a sample, and it can be pulled back up. And then we'll take those microbes back to our lab for studying.
Yep, and so these crazy tools that we use here on Earth are actually used on Mars right now on the Mars rover. So this is a picture of a mole that's on the Mars rover that is used to collect sediment from the subsurface layers of Mars. Unfortunately, this ice mole stopped working this year, but when we're starting to think about these icy moons, we may see rovers for Enceladus have a similar approach of an ice mole as what Teresa was just explaining. So by expanding our technology on these ice sheets and glaciers here on Earth to get at those salty brines, we can start to develop actual technology that can be sent out to these icy moons to then sample microbes if we find them. And so with that, we're going to go ahead and take you on a sampling trip today to a subglacial habitat and what it looks like when the ice mole is sampling from those subglacial habitats. So we're going to take you on a Jello sampling trip, unfortunately, but it's still going to be super exciting. And so before we go anywhere, we wanted to take a break for some questions and also let you know if you wanted to reach out to Teresa and I, here are our emails. Feel free to reach out to us with any questions.
Yeah, questions not just about what's in this presentation, but about astrobiology in general and how you can get involved with it and think about potentially having a career in astrobiology yourself.
Thank you so much, Taylor and Teresa. We will go ahead and leave that up for just a little bit in case anyone wants to collect that information and write down your email addresses. We did have a couple of questions that came in, and I would encourage people that are watching right now to keep typing them in the comments. We will share them so that they can be answered. The first question that we got was from Stacy, and Stacy's wondering if microbes are living organisms.
Yes, they are. The microbes beneath the ice are actively growing and living.
So the next one that came in, I guess, is from Angela. And Angela's wondering if there are analogs on Earth too that are actually super hot instead of super cold.
Yeah, so that's a great question. So there are analogs to places like Venus where it's super hot, such as what we may see in Yellowstone. Some of the hot springs in Yellowstone are lab studies as a way to understand how life may have evolved on Earth in these really hot time periods. And maybe a way to understand how life exists at extremely high temperatures on other planets, if life were to exist. Did you have anything else to add to that?
We also have places on Earth with really high pressure too, like beneath the ocean. So we have a spectrum of temperatures, pH, pressures. Earth actually has a lot of really cool ecosystems that we can use as analogs for different types of places on other worlds.
Awesome. Thank you. Casey also had a question too. Casey was wondering how deep is the ice? And I'm going to assume that Casey's maybe thinking about in the environments that you're sampling, maybe not necessarily on Enceladus.
Yeah, so that's a great question as well. So it depends. When you're thinking about mountain glaciers, it'll be not as deep as an ice sheet. So ice sheets are much thicker, and it takes more time to burrow through those larger, thicker ice sheets. So that can be, I think, like a couple miles. I can't remember how thick the Antarctic ice sheet is, but it definitely is much thicker on an ice sheet compared to a mountain glacier, which are the things that I work on in mountains, in the Rocky Mountains of Wyoming. So there's definitely variation in the size of ice on Earth and the thickness of it. But when you go to Enceladus, there's a lot of research that's working on that right now using satellite imaging. So I'm sure there's some pretty concrete numbers out there. I just don't know them off of the top of my head. I'm sorry.
No?
Do you know?
No, I don't. I think one of the lakes is about 20 meters beneath the ice. I don't know what that is in feet, 60 feet.
Mm-hmm. No, I appreciate you using metric instead of imperial. So, Leslie is wondering if you know anything about whether or not there's actually snow on these icy planets and moons too, or is it all just solid ice water?
Yeah, that's so many great questions. I'm so excited. Yeah, so there's different types of snow that can be formed. So there can be like I think snow formed by carbon dioxide and methane at super cold temperatures, but we're not really sure if there's liquid, like classic water snow. I'm not sure, but I do know that there is some snow that happens on those planets that's as carbon dioxide and methane. And then on Enceladus, another thing that happens is there was that picture I showed of the geysers happening. So a really cool thing about that moon is that as the liquid oceans are moving around, the surface of it changes a lot through these geysers that then deposits new layers of ice. And so this was a recent finding on understanding the dynamics of Enceladus, because old icy planets and moons generally have a lot of craters. But an interesting thing they found about Enceladus was that it didn't have a lot of craters. So they realized that there was a lot of activity happening with that ice sheet and that snow happening on the surface. So that's a fun fact.
Yeah.
Do you have anything else?
It might be slightly off topic, but how Earth has a water cycle that ends up with evaporation and then precipitation of water rain. Other planets have all kinds of other cycles going, like on Venus, it rains acid.
Thank you. Yeah, and one of the talks we actually had yesterday was with a NASA scientist who had worked on the Cassini missions that studied the moons of Saturn. And so, if people are interested, they can go back and watch that video. Dr. Paul Romani was talking about Enceladus a little bit more and explaining what you're describing. So I think that a lot of the other questions that are coming in are a little bit more about your process and your techniques. So I'm wondering if we want to go ahead and do your sampling demonstration, and then we can come back for some more questions.
Yeah, that sounds great. Okay, so we're gonna transition into our field sampling expedition. So here we go. So,
And we'll talk you through the process of making a Jello mold like this yourself, so you can copy it at home.
all right, so we're gonna go ahead and introduce our tools before we take you to this crazy thing. So today we're going to be using a straw and then a plunger, or what are these called?
Baster?
A baster. Yes, I had a moment. So we're going to use a baster to then sample into this glacier. And we have seeded this glacier with some little pieces of rice in here as our microbes that we're going to target sampling today. So I'll let Teresa walk you through the dynamics of the glacier before we go ahead and take our samples with the ice mole.
Yeah, you can use either tool you want, but I'm going to be going with the straw. And this is a boba straw, so it's a little bit wider. So the flour is ice, and then these are, the flour is snow, and then these are different layers of ice, and then beneath it, we have the liquid layer containing the microbes. And is it too loud? You can, sorry. So to sample, we first stick it in, and when it's all the way at the bottom, we're going to cover it with our thumb so that the suction will keep the Jello inside while we pull it out. So let's go for it.
All right.
And you can watch it slowly descend. And the way you would make this is just to use different colors of Jello, and when each one solidifies, then you add the next one. All right, so we've hit bottom.
So now she's gonna cover the top with her thumb to create that negative pressure in the tube. And we're gonna slowly pull it out like the ice mole.
Get the container ready for putting our sample in.
All right. So we have our first sample. You might need to squeeze it. So yeah, sometimes it's a little hard to get out of the straw. So you have to like push it out.
Yeah.
All right.
All right, and we can use one of our tools to pick through what we've got. And it looks like we do have some microbes in here.
Yeah, if you wanna. So the rice grains are bacteria that we're trying to sample, and it looks like we got some. So now what we're gonna do is we have our sample from the field. Normally field work isn't this lucky. You don't ever go out and get it on the first try. So great work. Now we're gonna take these microbes, put them into our sampling tube to take them back to the lab. So
yeah, we would bring back tons and tons of samples from multiple sites on the glacier.
So now that we've used our small sampler, we hit gold, we got our microbes, then we would want to increase our sample size. So then we would move to a larger mole apparatus to get a larger sample, to get more microbes and potentially more types of microbes. So we've collected our sample, and now we're going to go in with the bigger one. So remember, this is our baster. You can get these for $1 at Cub Foods. And now I'm going to go into the same spot Teresa went, and I'm going to try and get more of those microbes. So one quick look. This is what our core looked like. All right. So going down
is just
slow.
our name for the tube that has a lot of layers in it.
All right. Hopefully we've got something here.
Is that visible? Should we rotate it?
Yeah. Can you guys see?
Yes, we can.
All right. Now I'm going to go slow. Looks like I'm losing some sample. All right, let's see what we got. All right, fieldwork is messy.
We wouldn't dump out our whole core just to get to the bottom, though. We would take it slice by slice.
Yep. All right, and then I'm going to use my tool. I'm going to look around in my sample, and we have ice and water, so we have lots of things in here. And it looks like I got some bacteria, and I got a lot of them. So I'm going to make sure I take all of those back to the lab so I can study all the different types of microbes there and how they work. So got this, ready to go.
That's great. So you're ready to take this all back to your lab. Thank you so much for showing us that. I hope some people will try and do that at home as well. I bet you it's a lot easier to work with ice than it is with Jell-O.
Yeah, yeah. We have lots of Jell-O now, so maybe we'll play with it.
Excellent. So the questions that are still remaining are a lot about your process. And one is about this idea when you go back to the lab. April's wondering, what media do you use for your cultures? She's curious about the microbes from under the ice.
I'll let Teresa do this since she had those beautiful pictures.
So those microbes that you saw pictured earlier were grown of two types of media. One of them was R2A, which is a media that has basically almost no nutrients, because theoretically these microbes are from an environment with very few nutrients. So we don't want to give them too many nutrients. It might freak them out and they won't grow. And then I also grew some of them with a media that more resembles marine water. So it's higher in salt because these lakes beneath the glaciers might have a higher salt concentration.
Okay. So Christine is wondering if you ever fall into a situation where ice will ever clog in your instruments, and maybe what do you do when that happens?
Yeah, that's a great question. So the way the ice bowl works is it burrows down with heat, so it's moving through the layers by melting the ice, and then it will take a sample. So it's not as perfect as, or it's not, what we have demonstrated today is not a perfect representation of how the ice mold works. So I'm not sure. I'm sure that there is probably problems that happen as the ice mold goes down, but I'm not so sure that it would be similar to taking a core of ice, like what we're demonstrating. This was the closest thing we could come to heating Jell-O and moving down to the bottom.
Jell-O is already squishy, so it definitely doesn't need to be heated.
Yeah.
So Gary's wondering if you look for astroviruses, if there's such a thing, and if there is such a thing, would that be dangerous?
Whoa. This is horrifying.
There are viruses that target only bacteria called phages, and they're pretty much present anywhere there's bacteria, right?
Yeah.
But they wouldn't do anything to humans. Now, the idea of whether there's a bacteria that could transfer to humans, not a bacteria, a virus that could transfer to humans, is something else to think about, because bacteriophages, the viruses that affect bacteria, don't transfer to humans. We only get ones that transfer from animals.
Mm-hmm. Yeah, and then another thing to consider is that we don't know if there is human. It would be hard to understand or predict if a virus would be able to infect a human, because these organisms may have independently evolved away from humans. So it's very hard to know if these viruses would be serious agents for diseases.
Yeah, I'm sure they've never seen a human sail all those feet under the Antarctic ice.
That makes sense. So Rachel's wondering what kind of microbes do you actually check for in your samples, and do you also check for evidence of microbes from the past?
Yeah. So some of the microbes that I study bacteria, archaea, fungi, and algae. So these are similar to things that you would find in a lake. So you could go outside and see a lot of these microorganisms in your backyard by just taking a scoop of pond water or at the bottom of a pond. So what some of the things that I work on is studying these organisms under these extreme conditions and understanding like the diversity of them and how the carbon cycling is affected by them. And so those are the main organisms that I work on right now with my research. And And most certainly, if you're going down through these layers, you will get some of those microbes or DNA from microbes that have been there for a while potentially, or have been there for a really long time and have been living for a very long time. There was a recent paper that came out that talked about microbes that have been living on the seafloor for a very long time. So it wouldn't surprise me to see some microbial activity from very prehistoric times still happening at slow rates.
As for the microbes beneath the Antarctic ice, that's pretty much a closed system, so new microbes wouldn't really be coming in. So those are the original microbes from when that ecosystem formed. And a lot of them are actually kind of similar to what we would see in a really salty ocean. So they might have evolved from ancient ocean microbes.
Excellent. So I think I have one final question that might be a good way to end our time today. And Haley's asking, if you could study a sample from any planet or moon, what would you choose?
Oh, Enceladus all the way.
I think I would pick Titan. It's a moon that has methane lakes, and that's really cool. Methane is a hydrocarbon, like gasoline. So to have methane cycling and raining out of the sky and gathering in lakes, that's just awesome. Plus we have microbes on Earth that eat hydrocarbons. So why not have them on Titan?
Yeah. Yeah, and isn't on Titan there's like a huge pressure, so water actually forms pebbles. So it's the water actually is like as dense as a pebble, but the methane is like a lake.
I don't know how the water acts on Titan, but I know that it's like the only moon that has an atmosphere.
Yeah. Thank you. Those are great choices and ones that we've talked about this weekend. So I hope that everyone will go back and look at some of the past videos from this past weekend, learn a little bit more about Enceladus and Titan, and also learn a little bit more about these processes that you've been talking about. So Taylor and Teresa, I want to thank you so much for being with us today. I want to thank our audience once again for joining us for Space Fest all weekend. We have lots of exciting things coming up in the coming weeks. And one of the things that we're most excited about is that the Perseverance rover is going to be landing on the surface of Mars. And that brings us such exciting opportunities for research because those samples will actually come back to Earth where we can study them in labs all over the world. And so we're going to be holding a special star party in February, and I will be posting that to into the comments here so that you can go ahead and register for that. It's a free virtual star party. We'll be looking at the night sky, and we'll also be talking a little bit more about the Perseverance rover and that exciting research. So thank you once again. That's a wrap on Space Fest 2021. We want to thank you all for being here this afternoon. Have a wonderful day.
Thank you.